Good afternoon, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Deploying an NGS LIMS for Complex Research and Clinical Environments, and our sponsor is Sapio Sciences. Our panelists today are Caitlin Vaness, Project Manager of Sample Receiving and LIMS Development at Memorial Sloan Kettering Cancer Center's Center for Molecular Oncology and Integrated Genomics Operation, and Kevin Kramer, CEO of Sapio Sciences. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box on the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask the panelists questions after the presentations have concluded. Ms. Vaness, please go ahead.
Hi, everyone. How are you? Just give me 2 seconds to get set up here. Okay. So again, like Christy said, my name is Caitlin Vaness. I work on the Sapio exemplar LIMS system, one of 2 iterations that is purchased by Memorial Sloan Kettering Cancer Center. I'm going to talk a little bit today first about the different system specifications. Like I said, we have 2 versions of the exemplar LIMS in-house. I'm going to talk briefly on the structure of the 2 different departments that utilize LIMS systems, and then I'm going to delve a little bit deeper into the 2 different core processes and the differences between the research and the clinical system. The bulk of my time is going to be spent discussing how we have successfully customized Sapio's exemplar LIMS system to fit our specific research needs. And then lastly, I'm going to briefly review how the LIMS is integrated with a number of different interfaces across all of MSKCC and how we can both push information into the LIMS and receive it from the LIMS in a really streamlined fashion. So in brief, we have 2 systems here. The research exemplar LIMS system, we are currently operating on 7.11. We do have plans to upgrade to 8.0. We haven't implemented this yet, but the tablet support feature is a really large driving factor in our decision to upgrade to 8.0. Again, this is just for the research side. We do have the document search enabled. We also have purchased the dashboard module and the collaboration module that we utilize. The clinical exemplar LIMS system is running on version 6.33, and the exemplar LIMS system is validated by New York State Department of Health. For those of you in clinical environments, the lab itself is CLIA certified, and everything that it uses is also CLIA certified, but in addition to that, the LIMS system is also vetted by the New York State Department of Health, which was a very rigorous undertaking. And so upgrading the software system to a newer version is a challenge. To go into the structure of the core and the history of Memorial Sloan Kettering, it's important to understand that there's 2 primary entities within Memorial Sloan Kettering Cancer Center. We have the hospital itself, which runs clinical trials and has clinician-based research and is primarily focused on understanding human tumors. Then we also have a research arm that is Sloan Kettering Institute. And here we do all of our academic research. This is where we incorporate different animal models, and we understand a little bit more into the workings of basic science and how we apply that to cancer research. So the core facility that I actually work for is part of a research center arm. The arm is the Kravitz Center for Molecular Oncology. There's 5 main parts within this research center. So I actually work under the Integrated Genomics Operation, also known as IGO. And IGO itself is a core facility. So we process the physical specimens. we not only handle samples that come in through this Center for Molecular Oncology, we also handle a variety of samples from across all of Sloan Kettering Institute, from mouse, Drosophila, yeast, other species as well. And so we really have to be able to be flexible enough to handle anything. But as part of the Center for Molecular Oncology, we also have a bioinformatics core. We have a group called Knowledge Systems that run the CBio portal. Some of you may have heard of this. We have an innovation department that is really responsible for making sure that we're on the cutting edge, that we stay relevant, that we're ensuring that the methodologies that we use are the most current and up-to-date. And then we have another department that handles sort of higher-level computational oncology and really developing correlative studies and trying to understand the mechanisms behind cancer and making it into more than just data output, really trying to make the data into something that is understandable to a human. So we have a couple major areas that we do work with the Center for Molecular Oncology. The first is retrospective sequencing of human tumors. We do this through a number of methods, but specifically we tend to utilize capture panels, so either custom designed capture panels or the adjuvant whole exome panel. We also do a number of transcriptome analyses through a variety of RNA sequencing panels. And we do a lot of work, and we're getting more and more into methylation sequencing as a form of epigenetic analysis. We also do a number of prospective studies on both cell lines and organoids. So we've done CRISPR work, shRNA screens, different drug screens, and then we also work a little bit in liquid biopsy setups. So the important thing to note about all of the work that we do in the Center for Molecular Oncology is that none of it actually affects patient outcome. Everything that we do is after the fact. So the core facility itself is broken down. Our director is Dr. Anya Ciale. We have 19 wet lab technicians and 27 total staff. We have 8 liquid handling robots. We have 6 Illumina HiSeq instruments, 2 of which are the 4000. 3 of them are either 2500 or 3000, depending on the version. And then we have 1 2000 that is left. We have 3 Illumina MiSeqs, 1 Bio-Rad digital PCR instrument. We have Sanger, we have 2 Sanger instruments, and then we have NanoString that we run as well. Now, the counterpart to our department within the hospital is the MSKCC Diagnostic Laboratory. And as part of their diagnostic service, they have a lab that is Diagnostic Molecular Pathology, also known as DMP. This is a CLIA-certified lab. So all of the samples that are processed through MDS and the DMP are actually used to affect patient treatment, to enroll people in clinical trials. This is active work on patients in real time. And the LIMS instance used by this lab is approved by the New York State Department of Health and is subject to their strict and rigorous guidelines and checks. They do have a number of workflows that are both NGS and non-NGS platform-based. They're listed here. And then they also have several workflows that interact directly with their three-dimensional storage system, the ASM, which is used for DNA storage, and the SAM, which is used for RNA storage. Currently, to date, they have processed 11,000 patient pairs through their Impact, which is our clinical version of a capture panel used in the lab. On the research side, our volume so far this year, we've processed just over 19,000 unique samples. Some samples go into several processes, and I'll discuss that in a minute. We are on pace to double our volume from 2015, which is pretty remarkable. You can see that within our LIMS, right now we have 39,170 unique samples, so we're just about 20,000, and that's just this year. So we're really rapidly evolving and rapidly growing, and it's a testament to Sapio's system that we've been able to remain as flexible and really manage this huge amount of samples as they are processed through the core facility. Within the LIMS, we have them broken down into projects that can contain multiple requests. So we have just over 1,300 projects and 2,700 requests within those projects. So the LIMS in the research lab began when we needed to try to find a system that would be able to handle both samples in the clinical lab and samples in the research lab. And so after a number of exploratory trials, MSKCC decided to go with Sapio because we knew that they could really handle a variety of samples through a variety of workflows, and they had the option of having clinical validation that was very important for us. So in 2014, we purchased the two systems, and the idea was to try to share workflows, to share experiences, and we set up the research LIMS similarly to the way that the LIMS was set up in the diagnostic molecular pathology group, where samples were obtained in the clinic, they were paired in a tumor normal fashion, they were extracted, they underwent library prep and then capture, and then they were pooled and normalized and sequenced. And this really went through one process, start to finish. Multiple workflows, but one general process. And so we tried to apply the same idea to the research core facility. And the reason we did this is because a number of our processes are very similar. So we have two institutionally available custom designed nibbles and bait sets. The first is impact, and this panel is designed in such a fashion that it covers 410 known cancer genes that are commonly mutated in solid tumors. And this panel is also used in the diagnostic molecular pathology lab as their clinically validated test. Then we also have a panel designed for liquid tumors called HemePact, and this is 585 commonly mutated blood cancer genes. This panel is only available in the research lab currently. There is plans to very soon implement this in the clinical lab as well. And then we have other capture panels that are purchased by investigators or departments to cover specific aims. So because we have this very similar pathway, we thought that it would be very beneficial if we had a LIMS system that could sort of communicate back and forth and we could share different workflows. But as many of you know, research just isn't always so simple. So in the research lab, we will often go back and forth and really try to push things through even when in a clinical lab they would be failing. So for us, the ability to remain as flexible as possible was a huge hurdle to overcome when it came to customizing our LIMS system. So the research LIMS, the timeline, we purchased the system in October 2013. We purchased two systems, one for the DMP and one for the research lab. And then in January 2014, we started the custom design and really tried to make it work for everyone. We did have some research lab specifications, but we really tried to keep the systems as similar as possible. So in July 2014, we went live in production in the research lab, and extraction sequencing was one process. So we tested this out over the summer. We had a number of issues, and we realized that in order to really be as flexible as we needed to be in the research setting, that we just had to sort of rethink the way that we were processing the samples through the LIMS. So we actually scaled back, we took the system offline, and then we went online again in October 2014. And since then, we've been running relatively smoothly with some updates that we did in 2014 in December. And then again, when we upgraded to 7.0 in April 2015, that's when we did our second round of major updates that really tried to blend the flexibility and the rigidity as perfectly as possible to ensure that samples would move smoothly through the system, they wouldn't get lost, but that would allow us to have decision points along the way to decide on a very unique sample-by-sample basis how a sample would be processed through the LIMS. So to give you a little snapshot of what we currently have in our research LIMS, We have pathology workflows and extraction workflows all the way through to Illumina sequencing and MySeq workflow. We have workflows to handle nanostring and digital PCR. Our primary aim is sample tracking and project information, but we have a number of different things to interact with the LIMS to get information that we've recorded back out. So we have 42 sample processing workflows, and these are workflows that are actually linked together in different processes. We also have 6 non-processing workflows. These are workflows that we utilize to receive samples into the system, to move the samples through different container assignments, or to just add information to different projects or requests. We also have 39 active processes that we're currently using. We have 89 defined processes, but a number of these are processes that we no longer use, but we don't want to delete just in case we have to revisit them. And then one really big caveat to the way our system works compared to a clinical system is that all the research LIMS work is driven by an investigator request. So this means that samples, when they come into us, are sort of grouped together in these projects, and it really drives how the samples move through our system. So how do samples get into our LIMS? Initially, samples are collected by the investigator, and they fill out, currently they're filling out an Excel sheet. We're shifting into having them fill out a web form. The web form is going to be great. It will give investigators instantaneous feedback as to the validity and the completion of their sample set. Then when they bring the samples physically to us, they're imported in the LIMS, and we check them into their first process. So this is what the sample web form will look like. It, as you can see, is very driven by the type of sample that is being submitted and the type of process to which that sample will go to. And then once their selection is chosen, they will have a form that is very explicitly saying which fields are required based on the sample type that they've chosen. And then they're imported in the LIMS system under this hierarchy of project, then request, and sample. And one piece of information that is important for us is that an extraction request and a sequencing request are 2 separate entities, but they may contain the same samples. So a project that has 2 samples for extraction may go on to have 1 or 2 of those samples go into a sequencing request. And for us, this was a really large piece of customization that we had to work out with Sapio, and they came up with a phenomenal solution for us to allow us to retain the patient or the parent sample ID as it moves forward. So if sample 2 of a request fails, when they move forward into request 01234B, you'll see that the individual sample identifiers of 1 and 3 are retained. And then if we are in a situation where we're combining multiple requests, we need to make sure that there is still unique information in that 3rd request. So Sapio and we came together to come up with a really novel solution to help us with this interesting problem that wouldn't occur in a setting that isn't research-based. One thing that we've worked out recently is we decided to build into different quality control workflows this idea of a decision point. So we have 2 quality control workflows that we utilize, one for nucleic acids, and we utilize this when nucleic acids are coming into the core or after an extraction workflow has completed. And then we have another quality control workflow that we utilize for libraries or pools. And at the end of this QC, we have the ability to decide whether or not samples pass or fail. And it allows us to also get investigator feedback. So from our perspective, a sample may fail, but if an investigator knows that they may get some data out of this sample, even though we would not necessarily go forward with it, they may choose to override that decision. So we can record these pass-fail decision points in a really systematic fashion that allows us to see historically that we may have failed a sample, but the investigator decided to move it forward. So when we're doing our data QC, we can actually go back and refer to this and say, we knew that this probably wasn't going to be great at the outset. And really trying to record this information in a systematic fashion helps us ensure that we're giving people the best data that we can out of our sequencers. And then we also have, of course, these decision points where we can say, this one sample out of this sample set requires some additional work before we can move forward. So we've really incorporated into our QC this idea that a sample may not be destined for the same process, or a sample may need to be passed even though by our QC standards it fails. As part of this, we've increased our use of small standalone workflows. These are the two primary ones we use. So this allows us to split samples within a request into multiple processes. So if 3 of the 5 samples require some additional work, we can move those 3 into a different workflow, do some additional work on them, and then they'll all come back together in the same process. As part of that, we had to include this idea that samples could exist on the same plate, and we'd need to be able to call up this existing plate within the middle of a workflow. So again, Sapio helped us design this really great way to either call up an existing plate or create a new plate, depending on what is needed in a specific situation. So if you are entering the plate ID, it will tell you this plate ID already exists, or you can create a new plate with the same ID. And here you can see that we have the ability to see which samples are already on the plate and add our new samples to that same plate. One other bit of customization that we've been working really hard to implement across the board is the idea of robotic integration. So we use Beckman-Coulter robots to aliquot our samples onto plates, and they read a CSV. And instead of making the CSV manually, we are trying to have the LIMS create as many of these as possible. So once the samples are assigned to the plate and we've said how much material we are moving onto that plate, the LIMS will actually automatically generate for us the sheet that the robot needs to physically make the plate. So this sort of seamless movement from what used to be a very manual process has dramatically reduced our error rate. So the next thing I'm going to discuss is the way that the LIMS interfaces with different other systems, and I'm going to talk about this a little bit through the research first, and then mention some things about the way the clinical lab has their interface set up, and a little bit on how both systems utilize this final deposit called the CBio portal. So this slide is sort of just detailing how we have our LIMS instance set up both to receive information via an Excel or via a sample web form and have it then push information out into the pipeline, and how we can also access information about the samples through different RESTful service. So the first thing we have is a status board link out. While in the wet lab, the technicians are using the LIMS on a daily basis, we have other people that need to refer to information within the LIMS but don't necessarily need to be accessing the LIMS to get this information. So we've generated a status board that is both linked to the project in the LIMS but also pulls information from other locations. So this is an example of how we can display information from our LIMS to other people in a really integrated and dynamic fashion. And then the clinical lab has built a number of dashboards to allow people to review information based on what is in the LIMS without actually having to log into the LIMS to find the information. And they have this set up not only for their NGS platforms, but also for non-NGS services. Then one big thing we've accomplished this year is to have the LIMS completely integrate with our pipeline analysis. So after the samples are sequenced and we've passed them on for processing through the pipeline itself can get information from the LIMS that it needs to move the samples through. Here you can see we have our information about the sample type, the class, whether it's tumor or normal, how much input was put into library prep, how much the library was yield was, and what type of capture we have done. And this is really important as someone is going through and actually checking the different variants and checking information about how well the sample has performed without the information from the LIMS, it's a much more manual process, and we've tried to make it as streamlined as possible. So then the last place where our LIMS sort of really interfaces with a website is the CBio portal. So the CBio portal is a website that we have set up here at MSKCC, and it really contains information from both the clinical LIMS instance, the clinical lab, and the research lab, and it really is driven by the information that investigators are giving at the input for how samples are processed through, and then what the sequencing data that comes out of those samples is. So we can then query the system based on, projects, or based on different cancer types or mutation types. So based on how well the investigator has provided us with information up front, we can make this very integrated and very advanced place for anyone to come and query information about different tumor types, different mutation types, different cancer projects or studies. We have TCGA data, but this is our sort of ending point of where the LIMS finishes depositing all of the information. So to sum up, what we learned while integrating the LIMS system is it's best to start small with simple workflows to capture minimal information. And then it's really important to standardize small changes before incorporating new changes. When you live in a constant state of change, it's important that people feel comfortable with the change that's already occurred before you put a new change on top of it. And building robust workflows, and I feel like this is kind of the take-home message here, is that you can use the web tools within Sapio's LIMS system itself. You don't need programming experience to build really complex and complicated workflows. So our system is set up that I am the research side LIMS manager, and I work with programmers inside, the laboratory users, and the Sapio team to really accomplish everything that the lab needs to build this LIMS into a really robust system. So on that note, I'm going to just give a quick thank you to everyone at Sapio and everyone at MSKCC that has helped get the LIMS system up off the ground and really build it into the powerful system that it is today. So I will hand back to Christy, who will take over from there.
Thank you, Ms. Vaness. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We will conduct the Q&A portion of the webinar after the presentations have concluded. Next up is Mr. Kevin Kramer. Please go ahead. Kevin, please go ahead whenever you're ready.
Hi, good afternoon. This is Kevin, and first want to thank Kate for that nice presentation, giving a good comprehensive view of how they've used Exemplar there, and what a great job she's done as well in facilitating the successful rollout of Exemplar at Sloan Kettering. Oh, my screen's not changing, so there we go. Okay, so we're Sapio, a little bit of background on our company, and then we'll talk a little bit about our solution and how we're able to do an implementation that's complicated like Sloan Kettering. We're headquartered in Baltimore, Maryland. If you went to ASHG last October, we were right down the street from there. So this is Pratt Street, right off the Inner Harbor. We lease some space here on the 8th floor. And this is primarily our sales and marketing office, but we do have customer training and design meetings here on occasion as well. It's been a great location for us, and we're going to maintain this location for the foreseeable future. But, over the last several years, and thanks to our customers, we've experienced extremely rapid growth. So we needed a larger facility to house our development and consulting staff. So we acquired real estate just north of Baltimore, about an hour up in Pennsylvania, and we actually fully own this facility here. So this is really it's really a product of the success of Sapio and Exemplar. I'll point out that Sapio hasn't taken investment from venture capitalists, or even though we're contacted quite frequently to do so. So it's not artificial growth at Sapio where somebody handed us $20 million. This is all hard-earned through the success of Sapio and Exemplar, and through our customers, who've really enabled us to be successful with our solution. So we have extreme gratitude to them. This has been a really good location for us. We recruit heavily into our consulting and development staff, and the university systems around here are quite good. So, for example, we're going to Carnegie Mellon next week, and we're going to be interviewing a bunch of people there. We got Philadelphia with Ivy League Penn, where we have employees from, and to university, and even Penn State has a great computer science program. So we've been able to recruit and just be real successful in facilitating a top-notch team, which is really what we need here to be successful. And most of those people now report into this office here in PA. And it's also centrally located. I mean, between New Jersey, the NIH corridor, and then also up in Cambridge, there's a significant amount of pharma, biotech, and research going on at region. And we're very close to all those locations from here. LIMS aren't always the most popular things, so when people are saying good things about your solution, it's certainly notable, and we want to share that. So there's some representative quotes on Exemplar. I'll give you a minute to read these. And one of these actually is from Sloan Kettering, by the way, from the CLIA lab, so known as the DMP. So this is very important for us. There's the reason LIMS aren't popular is because there's been a lot of failed LIMS projects. Sometimes many years and millions of dollars are spent on projects that end up failing. And or the users just don't really want to use the system. It's too slow or not well designed. So for us, having successful customer implementations is what drives us here, and that drives the development of the solution to to have clean, easy to use design that performs well and is also flexible, because the LIMS market is quite diverse, and I'm going to talk about that some on the next slide. So what are some of the key attributes that enable, a kind of successful implementation like at Sloan, both on the clinical side and on the research side there? The first thing I would highlight, it seems like there's lag for my PowerPoint showing here. Okay. All right. So configuration. So of course, every product on the planet talks about how configurable they are, but of course the devil's in the details. And we've been at this quite a while. NGS is one of our fortes. We sell into all types of labs, so it's not just NGS. We do all types of implementations. Many are genetics and genomics based, but many are not as well. So we've done different types of NGS processes. But one of the reasons we can do these is the configurable nature of the product. And configuration takes many forms. It's configuring workflows is one of the key things, and I'll talk about that in a minute. But even configuring the user experience, what a user is going to see when they work in the solution is another part of it. Configuring the data model. This to us is a critical element of success, being able to configure the data model. And this is something you won't commonly see in a LIMS solution. This is something we had 8 years ago. So, we've had a lot of experience in refining and making this do what it needs to do in order to have successful projects. This is really a key aspect. I'm going to spend a little time on this point because Exemplar is a workflow-driven product, and what does that actually mean? If you go back 7 or 8 years and you ask people what they thought a LIMS did, you weren't typically going to give an answer that had anything to do with the, getting into detailed tracking of the processing of the samples through the laboratory. It usually was, general sample management, storage of samples, maybe tracking aliquoting or an extraction, but really not doing like a detailed type tracking like Sloan Kettering just showed, where they have 43 workflows, going through entire complicated pipelines. That really wasn't thought of something that was part of LIMS 8 years ago. But at the time, we actually introduced the workflow engine, and this workflow engine was in fact designed exactly to address that. So we wanted to do more than just manage samples in the freezer and shelf they were in. We wanted to track the processing details of the samples at a level that you could track in a clinical lab and be compliant. So the workflow engine came out 7 or 8 years ago. And the reason I think that's important is there's certainly some vendors that are now moving in this direction, but I think that having a vast amount of experience in doing these workflows, which we certainly have had in that time frame, has enabled us to understand several things. First off, what we need to do feature-wise in order to have the workflow be able to do what people need to do. And as I said, this is a great diversity of things, not just NGS pipelines, but other pipelines as well. And so making the workflow engine as flexible as possible and feature-rich as possible has been a real learning experience. So we've had 7 years of iterating on this workflow engine to make it more and more robust. The second thing is performance. We deal with the whole gauntlet of volumes. Kate's talking about tens of thousands of samples a year, but we have customers that might be dealing with millions of samples in a year. So, it could be a huge volume. We have some that might be doing tens of millions of samples per year. So this is a whole different tier, and you have to make a workflow engine that can actually scale to that. And we've had a lot of experience in refining and making this a scalable solution, not only to amount of data, but number of users. You might have noted that there's roughly 25 or 30 users using the LIMS at Sloan, actually more than that, I think, on the research side, and often on concurrently. So you have to have a system that's going to scale volume and concurrency as far as users. And we've had a lot of experience making the workflow engine scale. And this is where the lab techs are going to spend a large amount of their time, is in this. So the third part of it, and why this is important, having experience is important, is also in how you build these workflows. We're interested in being as efficient and fast as we can in building these workflows while not sacrificing functionality. So you still have a best-class experience. So what's important here is the tool that makes the tool. So if you think of the workflow engine as the tool, how do what tool do you use to make those workflows? So we've invested heavily in building out tools and capabilities around configurations that we can build really complicated workflows very fast that are, create a great user experience, will perform well, and are straightforward for the lab technicians to learn and work with. So, and I wouldn't always say having some experience is the best, the best thing. Sometimes a newer solution might have something novel, but I think in this case, actually, it requires a huge amount of experience to actually know what you need something like this to do. And I think the best way to get that is through working with a huge diversity of customers, which we have, and just implementing things and learning through that experience, and that's what we've done. So it's really a testament to our customers, as to how good this workflow engine has become. We have a lot of out-of-the-box. NGS is our largest business by far. So we've built up a huge domain knowledge in this area. We had NGS customers 6 years ago before it was in vogue as it is right now, as you can see by the attendance at this webinar. So we've built up a huge amount of domain knowledge here, and we know these workflows well. We know how the system should work, so our experience pays off, and also we have a lot of out-of-the-box capabilities with our baseline NGS system. So we do this thing called automated process management. This is something Kate described very well. But basically, systems are comprised of workflows. But what you really need to make the system robust is to actually have it that it can tell you, what's going on at the lab at any particular time. This actually can be quite complicated, especially if you want to have full pipelining. So if samples come in the lab and you want to route them into 3 or 4 or 5 different pipelines in parallel, actually tracking that is quite complicated. And this is what we're talking about with this automated process management that we can do with Exemplar. And then we can present dashboards of this information that the lab technicians can work from. So this is something we think is a distinguishing factor for Exemplar, that we can just configure these pipelines, and then Exemplar takes over with this automated process management, basically pushing the samples through the workflows for you automatically. We also do, we've had materials management for years, so we can track your consumables. In system, we track volume on hand, we have reorder alerts, et cetera. This is all configuration in the system now and quite robust. We can also manage your instruments, and we integrate with instruments as well. So we can manage them as far as maintenance is concerned, and then we integrate with instruments, and we have a lot of out-of-the-box integration that we can just provide. It's just given to you. For example, with a lot of genomics QC instruments, we already have out-of-the-box integration. The sequencing instruments, we have integration. One of the key things we think is important is not just to have the LIMS, sit there because you think you have to have it and collecting data that you're not going to make much use of. So for us, we want to make it very easy for you to actually utilize that data in meaningful ways, particularly for management and executive and lab management who are more directly involved, and people like Kate who want to know what's going on at the lab or what's historically going on. So we've worked really hard to make searching as powerful and easy to use as possible in the system, and this allows you to get at the data that you need to get at, and then either analyze it in Exemplar or export it and use it in tools, or use it through integration. As you saw in Kate's slides, they have dashboards that enable them to easily, through our API, pull out all this key information from Exemplar and do some really powerful analytics for their researchers as well. And they're also pushing data into Exemplar using the API as well. So they're leveraging searching tools for that. They're leveraging the API. And we also have report building tools right in Exemplar that make it easy to do these things. We also recently introduced, I just want to mention this, we introduced an ELN recently. So this is a different module than the workflow engine, so I'll try to distinguish what the two things are. So the workflow engine is very excellent at tracking these rote, repeatable process. So you want to step-by-step kind of go through in a certain sequence and complete something through the lab. And you want to do that in a very predictable way to ensure quality and throughput, et cetera. What we felt like we needed to fill as far as a gap is to some scientists who are doing more ad hoc things. So scientists might come to work, and on any given day they might be doing something totally unique, but they need to track it. They want to track it for IP protection because if they're going to make actually some amazing discovery, they want to be able to patent it. They have to provide evidence of what they've done. That's interesting. What to us is a little more interesting is that allowing them to build out a notebook, but then with our searching tools enables people to search across everything that's been done by the scientists across their entire company. So if you imagine you have hundreds or thousands of scientists, building out their notebooks every day, saying, I worked with these samples, I did flow cytometry on them, and various analysis, and this is all in notebooks across the system. And then this is all searchable, across the entire organization. To us, this is a powerful concept for discovery. And our motto is your partner in discovery, so this is something we're quite excited about. And we're trying to address several what we consider deficits of the current ELNs on the market. Ease of use is one of them. ELNs can be quite complicated and to such a degree that scientists actually don't want to use them. So we worked very hard on the design of this to make it clean and easy to use. Also performance, it needs to be able to scale, and scalability has been a problem with ELNs, so we're working hard on that to make sure the system is responsive for users. We also want to support multi-user notebooks, and what this means is that if a scientist is working on a notebook experiment, that other scientists can come in and work on it exactly the same time. There's no, well, I'm in here, now I have to exit, and someone else can come in now. We believe in collaboration is the best way for scientists to discover things. So we've made the ELN a multi-user capable at the notebook level, so multiple people can be working in the same notebook at the same exact time. And I could go on. There's a huge number of features in this ELN that we think are differentiators, and if you're interested in that part of the product, we could certainly do a demo for you at some point. So in addition to the reporting, we also believe that visuals are obviously important. Kate showed some significantly useful visuals, I think, for scientists to use, and those were created by them for their environment. But we also have dashboards in the product. So we have really key dashboards. I mentioned one, which is kind of a work queue dashboard that the lab technicians use, but we have other dashboards as well that allow you to see key metrics in the laboratory and look over any time frame that you want. And then there's some other powerful features in there in the dashboard package that really, we think, are differentiators for allowing you to get to the information in Exemplar. So we kind of are making Exemplar, I shouldn't say kind of, we really made it into a business intelligence type system. So we built into the system. You don't need to use some third-party product and then, connect it somehow to the system. It's built right into the system and is very, very easy to use. So this is kind of a lab intelligence system. It's not just a LIMS. It's more than that. You're collecting data. Now you can get actionable intelligence from it using our dashboard capability. And lastly, just talking about architecture, if you're going to invest in a LIMS, it's important to be looking long-term. So, what is long-term? In my mind, you'd be thinking at least 10 years, maybe longer, maybe 15 or 20 years. So we invested heavily in creating this browser-based application, and that's what we've had for years now. And it's a true web-based application. That means that there's no software to be installed on your client machines. And it supports all the major browsers. So we aren't saying you have to run on Chrome or you have to run on Safari. We support all major browsers that are available. And it also runs on tablets. And he highlighted this. This is a very hard challenge, by the way, and I think this is why you don't usually see LIMS vendors saying they'll run on tablets. It's not easy to actually run on a tablet. There's very many challenges there. But in fact, Exemplar is usable and works well on a tablet. And again, iPad, Androids, what have you. And this makes sense in the lab to me because lab technicians aren't just sitting in, like me, sitting at my desk all day. They're moving around. They're going over here to the thermocycler, and then they're going over here to the sequencer, and then they're going to the robot, and then they're going somewhere else. They're going to a freezer. Really, the LIMS should be able to go with them. So a tablet makes a lot of sense, I think, for lab technicians because of the way they move around the laboratory, and we have it, and it works. So we think that's an important feature. It's running in the browser still, so we just adapt accordingly to the form factor that we're working on. And Exemplar is also multi-tenant, so this means that you can actually have many LIMS running on one deployment, and you can spin up new labs or applications on Exemplar quite easily through that architecture. Also, we cloud host, so if somebody wants us to host the software for them, we can do that. We use Amazon as our hosting provider, and because it's all browser-based, we can do that quite easily. And I think if there's anything else here I want to highlight, I think that's it. So I think that's the end of my presentation. So I'll turn it over to Christy.
Thank you, Mr. Kramer. Before we begin the Q&A, we'd like to ask attendees to give us their feedback by taking our exit survey after the webinar. Joining us for the Q&A today will be Aaron Gabo, Lead Engineer for the MSKCC Integrated Genomics Operation, and Mustafa Syed, Lead Engineer for the MSKCC Clinical Setup. So our first question is actually for Caitlin, Aaron, and Mustafa. Could you discuss how many dedicated developers it took to develop the first working version of the clinical and research LIMS instances? For example, it looked like the first deployment phase of Exemplar in the research lab took about 5 months. Excluding external dashboards, have you had internal programmers working on Exemplar from the beginning, and did they need to go to Sapio training?
That's a pretty complicated question. So let's start with the first part, which is the first 5 months, there was actually, I don't believe, any MSK programmers working on the research side. It was just integration. Basically, it was some lab staff were working with Sapio directly. Mustafa, you were working on the clinical side. So in the beginning, the amount of support staff on the MSKCC side was very minimal. Kevin, you can probably comment on whether or not there was some additional staff on your end. But I came on board in June of 2014 and did basically a really fast catch-up. I did not attend Sapio training. And then, Aaron, when did you start working?
Around the same time I came onto this project, initially part-time, then more towards full-time. And I've been the only developer working for the research side. Here, they have also used Sapio's development team for some of it. but I did not attend any training. They have documentation, and that was sufficient for me to get through needing to do the programming I've had to do for them.
On the clinical side, we started, I joined when we have some NGS workflows already in place, but after I went to one of these workshops, I felt like it's very helpful, and you can code a lot of plugins in-house and move the development work in a much faster pace. And especially when there are production issues, it's better to have a good knowledge of these plugins so that it can be patched real quickly.
Thank you. Caitlin, did you validate the system? And if yes, what were your parameters?
I'm not really sure what that question means.
I'm assuming it's for the clinical system, but we can move on if you're not comfortable answering it.
Again, I don't do anything with the clinical system. Mustafa can possibly answer that question.
Well, we did worked on a lot of test data on a development server, and we validated all the workflows. We make sure like there are no loopholes and there are no issues before we release these workflows into production. And another thing that is associated with production with the clinical system is audits, internal audits, audits of the servers and application, and we went through these two audits also.
Great, thank you. The next question is for Kevin. Does Sapio offer local hosting or a private cloud in addition to a multi-tenant cloud?
All customers. So, for example, we wouldn't host multiple customers typically in the same EC2 instance on Amazon. So usually people are getting their own dedicated instance. If we utilize the multi-tenancy, it's for that customer account. So they might have multiple labs or applications built on Exemplar, and then on one EC2 we might use the multi-tenancy aspect to address that. I can't say in the future we would never have a shared EC2 instance by multiple customers where we leverage the multi-tenancy, but currently that's not the way we do it. Everybody gets a dedicated instance of Exemplar.
Kevin, does Sapio provide help or support for labs that want to clinically validate their LIMS?
We do. So we have actually a partner that we work with, a company called Zifo Technologies, that they're expert in that and have worked with us collaboratively on doing these validations before. We're also in the process of providing some out-of-box validation scripts on some of the key workflows in the NGS system that we'll be able to offer for people to acquire if they want to use those as starting points.
The next question is back to Caitlin. How much LIMS experience did you have prior to your current role? And can someone with no experience in LIMS development take on the challenge of implementing Sapio LIMS systems in his or her lab?
That's a great question. So I came into this role having only some very simple database design experience using primarily FileMaker Pro. So I would say that very easily someone with little to no LIMS experience can walk in and using the developer tools that are already built into the system, create pretty robust workflows without any background in programming or LIMS experience.
Question for you again, Caitlin. How many people are currently using your LIMS, and how do you train everyone?
So there's 27 people in the lab. There's 19 technicians that actually use the LIMS to run samples through start to finish. And then there are additional people that need to just investigate the LIMS for information. We currently have 36 user licenses, and then we use API licenses to display information in other locations for people to just look at displayed information.
Using your current knowledge, Caitlin, is there anything that you would go back and do differently regarding the implementation?
The only thing that I have to say that has been a major drawback was the idea of having sample names as a, we rely on the investigators to provide us enough information to develop sample names, and we tried to likely include too much metadata within those names. So I would say if I had to go back and do it again, I would ensure that we had a unique identifier per sample that remains with the sample through its lifetime, that is not dependent on any investigator information.
Caitlin or Mustafa or Aaron, can you tell us how many people in the MSKCC lab are using Exemplar and tablets, and whether you see that number increasing in the future?
Well, right now, it's 0 because that is a feature that is with 8.0 and we're still on 7.1. We are looking to switch over to 8.0 for the tablet usage, and then that would be probably most of the technicians.
Ladies and gentlemen, as a reminder, if you'd like to ask a question, please type it into the Q&A panel on the right side of your screen, and when you click on Send To, please select All Panelists. We have no more questions in the queue, so we'd like to end the Q&A today. Thank you very much for your time. We'd like to thank our panelists, Caitlin Vaness, Kevin Kramer, Aaron Gabo, and Mustafa Syed, and our sponsor, Sapio Sciences. If we didn't have time to get to your question, we will try to have it answered directly afterward. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.